Hello, I, Ramanpreet Kaur, Joseph and Prag. We are going to presenting our research project presentation on gibberellin and their effect on vertical growth of dwarf pea plant for our Biology 111 section Spring 2024. Begin with the introduction. Gibberellin, a group of plant growth hormones or a plant growth regulators. They are important for the maturity and the growth of plants. This experiment compares the growth of dwarf pea plant. We have four experimental pea plant groups, three of which are little marvel, which are given the low GA, high GA and no GA. An additional group of the regular height alderman. Experimental duration is for 3 weeks, which was observed by our group. Talking about the background information, Pisum setiaworm is a scientific name of pea plants, which are commonly used in a research experiment due to their simple genomes and the quick growth. Gibberellic acid, which is a key component of the plant growth and development, which regulates the various processes such as flowering, stem elongation, and seed germination. Now come up with the research questions. This study aims to investigate the gibberellic acid impact on pea plant growth, especially focusing on the stem height as a mean to observe and verify its effects on stimulating plant growth.
So on to the methods. Material we need in this project is germinated little marvels pea seeds, germinated alderman pea seeds, high and low gibberellic acid and solutions, pots and potting soils, beakers, rulers, growth lights. And then the procedure part. First we have take the four group of pea plants which are high and low gibberellic acid little marvels, no gibberellic acid little marvels, aldermans. And after that we have to plant 15 plants per group, which are 3 pots per group. And after that water and administers gibberellic acid every Tuesday and Thursday. Furthermore, measure height of stem every Thursday for 3 weeks, and at last record result and compare groups. So way we eliminate bias is by providing equal light to every plant and consistently watering and gibberellic applications. After that, measured from by base every week, and also same temperature should be provided to every plant, same pots and potting mix, and at last no cross contamination should be there. At last, due to unforeseeable circumstances, data is supplied by instructor as data collection was deemed impossible.
And now onto the results. The table on the right shows the mean height of each pea plant group at the end of every week recorded in centimeters. As seen from the information on the table, the low and high gibberellic acid groups exhibited the most growth, surpassing the other groups, while the no gibberellic acid group grew the least out of every single one. Additionally, while the low and high gibberellic acid groups were very similar in growth patterns, the low gibberellic acid group has the overall greatest mean height by a very slight margin. Continuing the results, the graph on the bottom right shows the comparison of the total mean growth of each group of pea plants at the end of the allotted 3-week time frame, measured in centimeters once again. The evidence from the graph shows that, as hypothesized, gibberellic acid does indeed have a significant impact on plant growth stem elongation. Additionally, these growth trend results are also in agreement with previous experiments, further supporting our conclusion. And lastly, these are our references.